Hi, this is the recording to accompany the slides for the second of two lectures on the cellular level of organization of the human body. So to start off with, I've got the overview of this lecture where I'm going to talk about DNA, its structure, and the process of replication to start off with, followed by the synthesis of protein in some more detail, and then continued on by talking about cell cycle, which includes the different phases of the cell cycle and how the cell cycle is controlled. And then I'm going to finish up by talking about stem cells briefly. So to start off with, when we talked about DNA before, you know that it's made out of nucleotides and it's organized into a double helix. So you've got two strands of DNA that are connected together in a double helix formation. A histone is a set of proteins with DNA wrapped around it along chromatin threads. And what you have is you have a nucleosome is defined as a single wrapped DNA histone complex. So the combination of the DNA wrapped around together with chromatin threads. And then you've got chromatin threads composed of DNA and their associated proteins. And then you've got a big chromosome that includes DNA and the associated proteins. And chromosomes is a condensed or tightly wound form of chromatin. So when you look at the structure of the chromosome or the DNA complex, what you can see here is you've got these multiple nucleosomes that appear like a beaded necklace, where the string is the DNA and the beads are the associated protein histones, right? And so they get wrapped around and wound really tightly to actually end up with that chromosome structure. One thing that DNA does is it replicates, it divides, it copies itself, right? So DNA replication is defined as the copying of DNA that has to occur before cell division occur. So cell division is when cells split themselves, divide themselves to produce two new identical daughter cells. Some cells divide very quickly. So your skin cells divide very, very quickly. Your hair cells divide quickly, while other cells don't divide as quickly. So some of the cells that don't divide as quickly are nerve cells, skeletal muscle cells, cardiac muscle cells, bones, and so on. When we look at the structure of the DNA molecule, what we can see is that we've got a double strand, right? Two strand of DNA nucleotides that's bound together with hydrogen bonds in a double helix, similar to a long ladder that's twisted up. So you've got this double helix right here. And you can see here that you've got one strand and another strand over here, and the two strands are bound together via their bases, right, with hydrogen bonds, right? So you've got the rungs of the ladder are formed by these bases joining together from strand 1 and strand 2. And in DNA, there are 4 different kinds of bases or nucleotides, and the things that differ are the nitrogenous bases. So the 4 types have got the 4 types of bases are the nucleotides that contain adenine, those that contain thymine, those that contain cytosine, and those that contain guanine. And we talked about base pairing before, and complementary base pairing means that if adenine is found on one strand, then it has to be linked to thymine on the other. So if you look at the picture down the bottom, what we can see here is you've got a red adenine always bound to a yellow thymine bound to a red adenine, and so on and so forth. And then if we have a cytosine, it's always bound to a guanine. So you've got here, if you have a blue guanine, it's always complementarily linked to a green cytosine, right? Green and blue, green and blue, green and blue. And that's also depicted in the diagram in the middle up there, where you have a guanine on one strand that's bound together with a cytosine on the second strand. So that's called complementary base pairing. So if you have a region of a DNA of one strand that has this sequence, then the second strand then has the sequence T, C, A, C, G, G, A. Right? If you look at it, A, adenine always bound to thymine, guanine to cytosine, thymine to adenine, guanine to cytosine, cytosine to guanine, cytosine to guanine, and thymine to adenine, right? So that's what complementary base pairing is all about. The process of DNA replication is depicted in this. So the first thing is it has to start, right? DNA replication has to start somewhere. And so there are a couple of enzymes that are involved in the DNA replication process. I'm going to just talk about two of them. And by the way, if you see a word and it has ASE or ACE at the end of it, chances are that in terms of anatomy and physiology, that it's an enzyme. So there is one enzyme called the helicase enzyme, and you can also see what the function of the enzyme by looking at the prefix, the start of the word. So the helicase enzyme untwists the double helix of the DNA, right? So helicase and other enzymes untwist the DNA at the replication fork over here, because you can see here on the other side of the helicase and enzymes, the DNA is still twisted into a double helix, but at the replication fork, it untwisted, right? And you've got the helicase over here. So what happens is when the helicase does its work, then you've got two separate strands, and each of those strands then become the template strand. So you've got original template DNA strand over here, and then you've also got another DNA template strand over there. So that's the initiation. The second part is elongation, where you have a complementary strand being built against the two original DNA templates, right? And that is helped by an enzyme called DNA polymerase. So, enzyme, we know it's an enzyme because it's got ASE. And what does the DNA polymerase do? It acts to form polymers of DNA, right? So again, the name is the function. So what happens is the DNA polymerase here and here forms two new strands of DNA against the two original template strands, so that you've got complementary strands, and you've got two separate DNA double helixes being formed, right? And then at some stage, you've got termination with the N. So it stopped when the two original strands are bound to the new complementary strand, and when you've got two new identical DNA molecules being formed, right? The term genome is the full complement of an organism's DNA, right? So the human genome is all the DNA that's found in the human body. And then the proteome is, again, the full complement of proteins that's found in an organism, right? So again, this is one of the functions of DNA. It can replicate. Another function will be put out in the next few slides. When we compare DNA with RNA, so deoxyribonucleic acid to ribonucleic acid, what we can see is that if you look at the diagrams down below, again, please do not memorize this, right? One of these diagrams is DNA, and the other one is RNA. And DNA is, the D in DNA is deoxy, right? So it's missing one oxygen molecule. So if you look at these two, the only difference is this portion of the molecule over here. And you can see that this is minus one oxygen, right, molecule. So this is DNA and this is RNA, right? Because it's got one less oxygen molecule. That's the difference. But a little difference on one molecule will lead to huge differences in their function. All right. So similarities between DNA and RNA. Each of the nucleotide in both DNA and RNA contain a sugar, phosphate, and nitrogenous base. So you've got here a sugar, a phosphate, and a nitrogenous base. And what happens is in both DNA and RNA, you've got long chains, right? And with the chains, they are connected through the backbone of alternating sugars and phosphate groups, right? So the chains, you've got sugar, phosphate, forming a chain of DNA and a chain of RNA. When we look at the differences between DNA and RNA, in terms of the length, DNA is long, whereas RNA is shorter, right? DNA is double-stranded, whereas RNA can be either single or double. The sugar in DNA is deoxyribose because it's missing an oxygen. The sugar in RNA is ribose, right? And the bases in DNA are adenine, thymine, guanine, cytosine, whereas the bases in RNA are adenine, uracil, guanine, and cytosine. So the difference in bases in DNA versus RNA is that DNA contains thymine, includes thymine, whereas RNA includes uracil in its place. When we talk about the next step, which is protein synthesis, we've got the term gene expression. So gene expression transforms the information coded in a gene. So a gene is defined as a functional portion of the DNA strand and transforms that information in that gene to a final product, so to a protein. And protein synthesis is composed of two different steps. The first step is transcription. And in transcription, what happens is DNA is used to synthesize a messenger RNA strand. So this is step one. In transcription, you've got a section of the DNA called a gene that is used as a template to actually synthesize a strand of messenger RNA. So you've got, this is transcription from DNA to messenger RNA. And then the messenger RNA is edited, exported out of the nucleus into the cytoplasm. You can see here in the middle picture where it undergoes the second step. And the second step occurs in the cytoplasm, inside the cell, outside the nucleus. And the second step is called translation. And in translation, what happens is the information that's coded in the messenger RNA is translated to addition of individual amino acids that are then combined together to form a polypeptide. And once the polypeptide assumes its final shape, it becomes a functional protein. And then so translation occurs in the rough ER with the help of ribosomes. And after the protein is made, It gets packaged into these transport vesicles to enter into the cis phase of the Golgi apparatus. Golgi apparatus processes it and packages it, and then groups it into different vesicles, secretory vesicles, where it can either be exported to outside the cell, or it can be integrated into the plasma membrane, depending on which protein we're talking about. So this is the overview of protein synthesis. I'm going to talk about next about the basics, right? So when we talk about the basics, what you can see here is that there are these groups of 3 nucleotides together, right? And these group of 3 continues through in terms of complementarity from DNA to RNA. And so the group of 3 in DNA, if you break up the DNA into specific groups of 3 base sequence, it's called a triplet, right? So this is made out of triplets. Whereas once we actually once we actually transcribe that into messenger RNA, then it becomes a codon. And then what happens here is you've got the messenger RNA here. So you've got codon 1, codon 2, codon 3. And what you can see here is this codon 1 over here, AUG, actually then is complementary to the anticodon of the specific transfer or tRNA that actually will bind to it. And you can see here that the anticodon of the transfer RNA is then linked to specific amino acids. So this UAC anticodon of this transfer RNA is linked to this amino acid. The AAA anticodon of this transfer RNA is linked to this specific, another specific amino acid. So when we look at it, a DNA triplet is complementary to its corresponding messenger RNA codon. A messenger RNA codon is complementary to its corresponding transfer RNA codon. And each transfer RNA anticodon can attach to a specific amino acid, right? When we're talking about complementarity between DNA and RNA, what we can see here is if you have DNA, if you have adenine on the strand of DNA, it is complementary to uracil or U on the RNA strand. If you have thymine on the DNA strand, it's then complementary to adenine or A on the RNA strand. If you have guanine on the DNA strand, then it's complementary to cytosine on the RNA strand. If you have cytosine on the DNA strand, then it's complementary to guanine on the RNA strand. And you can see this, you can confirm this by actually looking at the diagram above, right? So you've got it right there. So if you look at the template strand, so we want to compare the template strand, not the second strand. You can see here, this is complementary to that. A is complementary to U on the RNA strand, C complementary to G, G complementary to C, and so on and so forth. So when we look at the strand of DNA with the sequence AGTGCCT, the complementary strand of RNA is UCA CGGA, right? Following the rules outlined over there. So let's talk about the first process of protein synthesis, which is transcription. So creating a strand of RNA, messenger RNA, from a complementary strand of DNA. So the first part is the start or initiation. So you've got a DNA sequence called a promoter sequence that triggers the start of transcription, right? So the initiation phase happens over here. And then the second phase is the elongation phase, where you have the creation of the new RNA transcript. And you can see here the enzyme that helps to create the RNA transcript is called RNA polymerase. Again, the name ASE enzyme, polymer means lots of blocks. And so the RNA polymerase enzyme helps to create, to form a long strand of RNA. And then at some point, it will reach the termination, it will stop because the RNA polymerase hits a stop codon, right? So typical stop codons are UAA, UAG, UGA, and then that will actually tell the RNA polymerase to stop making the RNA polymer, and to release the messenger RNA transcript at that point. After that, what happens is the messenger RNA transcript is edited. So what you have is you have the original transcript, the pre-mRNA transcript, and you can see here it's divided into exons and introns. So you've got exon 1 intron, exon 2 intron, exon 3. And what happens is you have something called the spliceosome, basically cutting it at each of these different sections. And when they cut it together, what happens is all the introns are combined together, right? And basically makes this the shape, and all the exons are joined together. So you can see here, exon 1, exon 2, exon 3 are joined together, and the exons then form the edited or spliced messenger RNA, and this is then the final copy of the messenger RNA that gets transported from the nucleus to the cytoplasm. And you've got here just a definition of introns. They're long regions of RNA that can be either non-coding or contain transcripts that give rise to different variations of the protein. So again, you can have the same DNA being transcribed into messenger RNA, and depending on the introns and exons that are combined together, or the sections that become introns and exons, will result in different types or different variations of a protein. So once the messenger RNA is edited and finalized, it gets transported from the nucleus to the cytoplasm, where it undergoes translation. So translation is the process of synthesizing a chain of amino acids called a polypeptide from the codons of the messenger RNA. And it involves the ribosomes that are found on the rough ER. So the first part is the initiation, where you've got the mRNA, and you can see here the ribosomes consist of a large subunit and a small subunit. And so you can see here that the messenger RNA binds to the smaller subunit, right? And then the large subunit then comes together towards the small subunit. And so what you see here is you have the complete ribosome with the large as well as the small subunits together. And then step 2 is when the polypeptide is built, elongation. And so you've got specific transfer RNA actually that that are complementary to the codon of the specific transfer RNA that are complementary to the codons of the messenger RNA being bound together, and then the transfer RNA is attached to specific amino acids, and then what happens is then the amino acids are joined together to form a polypeptide chain, right? So you've got these amino acids being joined together, and then at the end of the messenger RNA, you've got a termination or a stop codon reached, and then that actually tells the translation process that it's done. The polypeptide is completed, and then it's released from the ribosomal subunit. So again, amino acids are joined together by the peptide bond. So this is amino acid 1, and this is amino acid 2 on the right. So when we look at the overview, what you can see here is that when with the synthesis of polypeptides, it starts in the nucleus with transcription, where DNA is transcribed into a messenger RNA. And then you've got the editing or splicing of the messenger RNA into its final form. And then the messenger RNA leaves the nucleus to enter into the cytoplasm, where it undergoes the second process of translation, where you've got a individual amino acids being brought together by the transfer RNA to form a polypeptide. And then the polypeptides can then join together to form the complete protein molecule. So at the moment, we've got polypeptides. How do you go from a polypeptide to a protein? Right? A polypeptide is just a string of amino acids. A protein is a functional polypeptides. So as you know, most proteins have got complex structure. So you've got the primary structure, which is what is made by the messenger RNA and the transfer RNA, and that gets folded into the secondary structure, the tertiary structure, and if it involves more than one polypeptide chain, you've got the quaternary structure as well. So what happens is that process can occur with the help of cytosomes, enzymes in cytosomes. If the proteins are not folded or they're folded the wrong way, they're then broken down into proteasomes. Remember, proteasomes basically break down proteins or polypeptides into its individual amino acid components, right? And the word proteome is defined as all proteins that are synthesized by the cell. The next thing that I'm going to talk about is the cell cycle. So cell cycle, as it goes as it indicates, it goes round and around. So it's a sequence of events in the life of a cell from the moment it is created to at the end of a previous cycle of cell division until it divides itself to generate 2 new cells. There are 2 main phases of the cell cycle. The first one is called interphase, and interphase includes G0, G1, S, and G2 phases. The second phase is called mitosis, and mitosis is also referred to as the M phase, and it includes, the M phase, and the M or mitotic phase includes mitosis, which is the division of the nucleus, as well as cytokinesis, which is the division of everything else in the cell in order to create 2 identical cells. So you see here in interphase, you've got these different phases and what occurs in those phases. Most cells are in interphase most of the time, and they're in the M phase only when they actually want to divide into 2 new cells. Some cells hang out in the G0 phase, because they're not actively dividing. So some cells that stay in the G0 cell phase are like bone cells, right? So they stay in the G0 phase for a long time because they don't divide for most of their life. When we talk about the M phase, there are structures that are present during the M phase. So the structures that are present during the M phase include centrioles. And so you can see here the centrioles are the points of origin of formation of new microtubules. You've got the centrosomes that hold the that are a pair of centrioles that are combined together. You've got the mitotic spindle. So you can see here, you've got the centrioles and centrosomes, and then you've got the microtubules that make up the mitotic spindle. And then you've got the cleavage furrow. So you can see over here, the cleavage furrow is basically when you've got a band of microfilaments that squeeze or contract the cell so that it splits in half during cytokinesis, right? So what happens is that cleavage furrow develops and develops and develops until you can imagine that it basically then splits the cell to form two new cells, right? During prophase, the two centrosomes begin to move towards different sides of the cell. So you've got one centrosome, second centrosome, move to different sides of the cell so that you can form the mitotic spindles by growing the microtubules. If that's all gibberish, hopefully the next couple of slides will actually explain it a little bit more. Before we talk about the process of mitosis, and remember, mitosis is just the division of the nuclear content, right? So the synthesis of a second set of nuclear content actually occurs during the S phase, right? So DNA replication occurs during the S phase. The mitotic phase is when the nucleus actually is split in to form two new nuclei. So normally, each human cells contain 46 chromosomes, which are divided into 23 homologous pairs of chromosomes. And after the S phase during interphase, cells contain double that. So cells contain 92 chromatids, which is 2 copies of each chromosome, right? When we're talking about chromosomes and chromatids, this is just an attempt to actually define the terms. So when we talk about homologous chromosome pairs, what we're talking about is the chromosomes that each person inherits from their parents. So you've got homologous chromosomes. You inherit maybe the red one from your mom and the blue one from your dad. When we talk about chromatids or sister chromatids, what we're talking about is the chromosome and its exact copy. So sister chromatids, you can see that this is identical to this, right? So you've got a double of the chromosome being formed during the S phase. And the blue one over here is similar to the blue one over there. And then the next term is kinetochore. So kinetochore is this, which is a protein complex that attaches the microtubules of the mitotic spindle. So you can see here, this is during prometaphase of the mitosis process. And then the centromere connects the identical chromosomes to each other. So the centromeres connect the pair of identical chromosomes to each other. So in the next slide, what I'm going to do is go through the different phases of mitosis. So an easy way to remember the sequence of mitosis is think about your cat peeing on the mat. So you've got prophase, metaphase, anaphase, telophase, right? So PMAT. And I know that some of you have covered this in much more detail in biology or in previous courses. This is about as much detail as you need to know in this course. So you've got prophase to start off the mitosis phase of the cell division, where you've got the chromosomes condense, become visible, nuclear envelope and nucleolus disappear, centromeres produce spindle fibers, start moving towards opposite poles, opposite ends of the cell, and then prometaphase is continuation of the condensation and visibility of chromosomes. Kinetochores appear, and then you've got the start of the mitotic spindle production. And then in the metaphase, what happens is the chromosomes are lined up in the metaphase portion, right? And then in anaphase, that's when the centromeres are split in two. So you can see here, you've got one set going to the right, and then another set going to the left. The spindle fibers start to make the cell more elongated. So you can see here the cell then starts to stretch. And then in telophase, what happens is you have the start of the formation of a new nuclear envelope around each set of chromosomes. The mitotic spindle breaks down and disappears, and then the spindle fibers continue to elongate the cell. And then the next part, after mitosis is completed, you've got cytokinesis, where you've got the cleavage furrow appearing, splitting the elongated cell to form two new daughter cells. I've also got this information in a different format on this slide. So again, depending on whether you like to follow this kind of format or the previous format, it's then up to you. The last thing I'm going to talk about is, oh, sorry, this is not the last thing. So another thing that I wanted to mention in terms of the cell cycle is, how does a cell know when to go to the next phase? And how does the cell know whether it's good enough at that phase? And so what you have is you have these checkpoints and you have these enzymes, right? So the movement of a cell from the G2 to the M phase, from the M phase to the G1 to the to the S phase to the G2 is very heavily regulated, and it is regulated by these checkpoints that act as a stop or go signal. And this is very important in order for cancers to be prevented, right? Because cancer occurs when cell division and cell growth is not restricted. So you've got these various different checkpoints. So you've got the G1 checkpoint over here, right? And at the G1 checkpoint occurs at the end of the G1 phase, and it basically ensures that the cells are ready to synthesize a new set of DNA, right? Because replication occurs in the S phase, and the cell needs to be ready for that. So it needs to gather up all the materials needed for a new set of DNA to be synthesized. And then there's another checkpoint at the G2, at the end of the G2 step of the cell cycle. And this checkpoint, G2 checkpoint, basically ensures that the cell is then ready to divide. The cell is then ready for mitosis. And then you've got the M checkpoint to actually ensure, or the metaphase checkpoint to ensure that the sister chromatids are properly arranged, attached to the microtubules, line up at the metaphase phase plate, so that the division of the nuclear content can occur. So you can see these checkpoints are actually important to ensure that two identical daughter cells are produced at the end of the day. And we've also got several different enzymes called kinase enzymes, which are cyclin and cyclin dependent kinases. And these are activating enzymes to actually spur or drive the cell through its various phases and to also progress past the checkpoints. So it's really quite a neat system of checks and balances to ensure that the cell is ready before it goes to the next phase. And then the cyclins and cyclin-dependent kinases actually help to move the cell along when it's in whatever phase that it is. Last thing I'm going to cover is stem cells. So there are different types of stem cells that are found in our body. So stem cells as a whole are unspecialized cells that can divide without limit to form either new stem cells or to differentiate to form different kinds of more specialized cells. So there's different types of stem cells. Or different types of cells, I should say. So the first one is the totipotent stem cell. And this is found in embryos. And the totipotent embryonic stem cells can differentiate or divide into any cell that's needed to for an organism to grow and divide. So the totipotent embryonic stem cells can divide and differentiate into different types of pluripotent stem cells. So you can see here pluripotent stem cells, there's three different types. There's the cells of the endoderm line, cells of the mesoderm line, and cells of the ectoderm line, right? And then what happens is that the pluripotent stem cells can then differentiate when they divide into different types of cells. So if you look over here, the endoderm line can differentiate into different types of multipotent stem cells, and they include cells of the lung and the pancreas. The pluripotent cells of the mesoderm line can differentiate into heart muscles and red blood cells, and then cells of the ectoderm line can differentiate into skin cells and neuron cells, right? So what you can see here is as it goes down the stem cell line, it becomes more specialized and less able to actually create a whole new human being, right? So the multipotent stem cell line, so for example, of the blood cells, so you've got over here, the multipotent hematopoietic, so hemato refers to blood cells, can only divide or differentiate into cells that are connected to do with the function of blood. So the multipotent hematopoietic stem cells can divide into form platelets or thrombocytes, can divide to form white blood cells, right? Can also divide to form red blood cells. But you can see here all of these all of these cells below the level of the multipotent stem cell lines are all cells to do with blood, right? And these are called oligopotent stem cells. So an oligopotent stem cells is limited to become one of a few different types. So you can see here the lymphoid progenitor can form natural killer cells or small lymphocytes, but cannot form the cells on the left-hand side of that particular diagram. And then you've got unipotent cells, which are these cells over here. They're fully specialized and can only reproduce to generate more of its own specific cell type. So the megakaryocyte, for example, can only divide and reproduce more megakaryocytes, right? The unipotent stem cell of a red blood cell can only divide and produce more red blood cells, cannot divide and produce more platelets, cannot divide and produce all these different types of white blood cells. So the uni means one, right? The unipotent stem cells can produce only that particular type of cell. How do cells become differentiated, or how do cells become specialized? So differentiation can cause changes to cell size, shape, metabolic activity, and overall function. So the question is, if all cells in the body contain the same full set of DNA, how does differentiation occur? So the DNA in your muscle cell is exactly identical to the DNA in your bone cell, is exactly identical to the DNA in your nerve cell. So how does the DNA in each of these specific cells know what it should differentiate into? The answer is that each cell type only reads the portions of DNA that are relevant to its specific or own function. So, even though all cells have the same identical DNA, the genetic expression of DNA is unique to each cell type. So it means that each cell, in each cell, only relevant genes, portions of DNA, will be turned on, while others are kept off to produce specific proteins, etc. Right? So, it's like you. You are the same person whether you're in school or whether you're at home or whether you're at store, but you do different things at different locations. So just because you're the same person doesn't mean you do exactly the same thing wherever you are. We do different things at different times of the day, at different locations, and so on and so forth. So the things that govern the on and off of these genes are called transcription proteins to either promote, turn transcription on, or inhibit, turn transcription off. So for example, these are transcription factors that bind to this specific portion of the DNA or this specific gene to actually turn it on. So once it gets turned on, then RNA polymerase binds to it, and then the process of generating a messenger RNA strand begin. So you can see that these transcription factors may be unique to this particular cell, whatever it is. In another cell, they may not have these transcription factors. And so if there's no transcription factors in another cell, then it won't actually activate the promoter. All right. So I hope that's a good enough explanation. If not, you can always ask me questions in class. Thank you.